In this latest installment of Deep Evolution News, microfossils from the earliest Cambrian reveal Polychaete goes lineage and detection of new complex organic molecules on Mars. Let's dive into it right away. Sometimes the biggest discoveries come from the tiniest of finds. The Zhangjiakou Lagerstätte in South China has been known for well over a decade to yield highly interesting fossil remains from the earliest stages of the Cambrian period. Researchers have now uncovered microfossils of the earliest known annelids, or ringed worms, a phylum that includes earthworms, ragworms and leeches. Two new species, Quanchuan P. vermis brevicrudis and Zhangjiakou vermis longicrudis, were described in intricate details and dated to the Fortunian age 535 million years ago. Now, unlike arthropods, annelids have relatively soft bodies, stiffened mostly by a hydroskeleton, and just have a thin outer cuticle made of collagen and usually no hard parts, or very few. This makes it challenging to confidently recognize any annelids because many identifying characteristics are often not preserved. Up until this point, the oldest known unambiguous annelids were up to 518 million years old, from later Cambrian deposits which all have Burgess shale type preservation. For instance, there is Polychaete, which displays full body tagmatization into different zones, which is in line with crown group annelids. There are other possible contenders like Yilingia, hailing all the way back to the Ediacaran period, but these all lack defining features and may have been stem group annelids at best. What makes the current site so special is its so-called Orston type preservation. Unlike the famous Cambrian Burgess shale, where organisms are preserved as flattened carbon films, at Zhangjiakou, tiny fossils have been preserved as three-dimensional infillings. The difference is somewhat comparable to preserving flattened dried plants as herbarium sheets and filling a tiny balloon with cement and then popping it, removing the outer layer. With the Orston type taphonomic process, there's no preservation of either internal organs or external features on the skin, but only the body's general outline in 3 dimensions. And despite being tiny, only a few millimeters in length, this kind of detailed infilling gave the researchers enough to go after to confidently identify these fossil species as annelids. The microfossils are elongate structures showing clear signs of segmentation and are contorted in various ways, meaning they were flexible in real life. They have repetitive sideways extensions that branch out at the ends. This conforms very closely to the body plan of Polychaete annelids, with their biramous appendages called parapodia, which are used in locomotion. The authors rule out that these could be remains of algae, because in that case you'd expect to see individual cells delineated, and the branching pattern would be more radial. The species are also clearly not lobopodians, the closest relatives to arthropods, that look quite similar to Polychaete annelids. Most importantly, because lobopod feet are never biramous. An important distinction between the two species discovered here lies in the length of the parapodia. Quanchuan P. vermis has relatively short limbs, similar to that of a benthic sediment-dwelling ragworm, whereas the other species, Zhangjiakou vermis, has very long limbs. This and the overall morphology of the latter suggests affinities, or in any case a very similar lifestyle, to modern-day Tomopteris, which is a free-swimming holopelagic annelid. The implications of this are huge. The presence of a fully formed representative of one animal phylum, in this case the Annelida, at the very beginning of the Cambrian means that this and others already existed in the preceding Ediacaran period. The absence from the fossil record of any traces of these ancestral lineages have more to do with preservation and relative rarity rather than complete absence. This is another confirmation that many animal phyla had already diverged well before then, and that the Cambrian explosion may in reality have been more of a Ediacaran crawl. An experiment performed by the Mars Curiosity rover on a carefully selected rock has revealed the greatest diversity of complex organic compounds found on the red planet so far, and this includes potential building blocks of life. The current conditions on Mars are known to be highly destructive to organic materials, but it was not always like that. During Mars's early warm period, billions of years ago, the planet hosted extensive lakes and river systems. In these wet environments, many layers of sediment were deposited over long stretches of time, often saturated with complex chemistry. Amongst these, certain clays, specifically dioctahedral smectites, acted as a chemical time capsule, shielding the organic matter it captured from Martian surface conditions as these became harsher over time. Curiosity did some onboard forensic chemistry on a target rock located in the Glentorridon region of the Gale crater, dating back about 3.5 billion years ago. The rock, named after famed pioneer of paleontology Mary Anning, is a fragment of these ancient layered deposits and therefore meticulously chosen for the experiments. In a first time ever, the rover used a cutting-edge technique called TMAH thermochemolysis, combining alkaline chemicals with heat to strip organic molecules from the rock's minerals. The experiment released a staggering amount of 20 different organic molecules, including aromatic compounds such as methyl benzoate. Of special interest are two ring molecules, nitrogen heterocycles, which are regarded as precursors to nucleic acids, and benzothiophene, a sulfur-bearing cousin of the same family. So the sands of time on Mars have successfully preserved a complex organic record despite billions of years of radiation and geological decay. The question remains if this was actually produced by life, or simply the result of complex primordial chemistry, perhaps supplied from the outside by meteorites and comets. In any case, we can now confidently say that if life ever did exist on Mars and its products were captured in accessible sediments, we should be able to recover that it actually did. Alright, that's it for this time around. Please keep an eye on my channel for the next video, subscribe if you haven't already, and hit the bell icon so you don't miss anything. A super thank you to all my Patreons for supporting me this month around. For now, stay sharp and stay evolved.